So good morning, everyone. Thank you very much for the introduction. Thank you very much, Ioannis, the organizer, all the entire team for having me here today. It's a pleasure to be with you. Oh my God, it's a big screen, absolutely a big screen. Yeah, I mean, first of all, congratulations, you are the survivor of all this. Yesterday was a beautiful evening, and I want to thank everybody for the interaction. It was a really lovely dinner altogether. All right, let me just get through this one. So, the whole point of my presentation today, and I do have to say that you are unfortunately a bit unlucky because the Chief Scientific Officer of Thermofisher Scientific, Karen Nelson, was supposed to be here. Unfortunately, she couldn't make it. That's why you get the second choice, but I'll try to do my best. It's all right. I'll try to do my best. But the point is, the whole thing, the reason why I want to share some of these thoughts and ideas with you, it's in fact about bridging and filling a gap. So I will tell you a little bit what we're doing at Thermofisher, how we enable people to fill some gap, and what was my personal history in trying to, in fact, fill a gap. So the whole point is trying to take ideas and bring them into the real world, so basically making products out of this idea. Now, before going there, just a few words about Thermofisher Scientific. And I mean, that's terrible. It means I'm old. I'm starting with the first piece of advice. So that's whenever you decide to make your own company, to go work for a corporation, whatever you want, small, large, medium, make sure that you understand what is the mission of the company that you either, building yourself or that you're going to work for. And in case of Thermofisher, our mission is to make the world healthier, cleaner, and safer. How do we do this one? And what does it mean? It feels like this, super fancy corporate, whatever, it's not. It's a real thing. And to be honest, it's a driver of what we do every day. We make the world healthier because, for example, in my personal case, I do oversee the oncology business, and we do product in diagnostic, or we do produce drugs that do impact the life of hundreds of thousands of patients every year. We make the world cleaner because we do have solution, for example, that make sure that the water stays always clean. We do have product that allow us to check for pollutants, and so that's definitely very impactful, especially in certain part of the world. We also make the world safer. For example, we have a large business in the agro field where we have a lot of products that are used to check for pesticides, for contamination of foods, and therefore we do have very tangible impact in all these three different subjects. Now, a few more maybe words about Thermofisher, also a few numbers just to give you the size and the scale of our company. We have been a Fortune 500 companies for many years now. I want to start by what probably is one of the most important number in this slide, and it's that we invest every year in R&D about $1.3 billion. It's a substantial amount of dollars in there. These investments are actually backing up the work of more than 7,000 scientists all over the globe. We do have R&D sites that are spanning all the way from Japan to California, and I will show you some of these places. We estimate for this year something around $44 billion in revenue, and we have about 120,000 colleagues that are working with us all over the globe. So we are one of the largest corporations, in fact, in the life science business. It does take a little bit of time to get from one to another. Let me try again. I mean, I'm sure that if you ever worked in a lab or if you ever hold a pipette in your life, you most likely have worked with any of these brand in here, from Invitrogen to Gibco. But probably what most of the people don't know is that one of our largest business, it's actually in the manufacturing and production of drug. So with Patheon, which was a $20 billion acquisition back a few years ago, we in fact produce a lot of API for some of the largest pharma. We are also very much in the business of clinical trials PPD, one of the most important CRO out there, one of the largest CRO out there. It was also a relatively recent acquisition. And this is really enabling us to cover a substantial, a very large, wide span of capabilities. Now, these were just a few words again about Thermofisher. Now maybe how do I got to this one, and how Thermofisher accelerate and enables transforming idea into solutions. That works. Probably. Now let's see. Not really work. Okay, so this must be a huge file. This is the typical corporate, beautiful, very clean pictures of, this could be any of you. Okay, let's start with the first disclaimer. I think this is very important. Of course, I'm an employee of Thermofisher Scientific. I also work as a venture partner with the XGen Venture Fund, as you have heard. I'm going to express some ideas and thoughts that are my personal idea. They should not be considered necessarily as the position of the company that I am associated with, and I am not endorsing or recommending or promoting any of the product of Thermofisher or any of the investment that XGen is doing through this event. So that's a very formal disclaimer. Okay, now let's go to the really important one, the one that you should probably remember. I'm just another dude, and there are many of me out there. You just walk out the door, there are a lot of executives from many large corporations. So I don't have the presumption in any way that I can tell you a story that you have never heard before. You probably heard it already in a way, and I am not endorsing, recommending, or promoting anything that has to do with my own career. So not necessarily do what I've done. Now, what is this all about? The point is that, and I strongly suggest, oh my God, this is the second piece of advice, so I'm definitely getting older. Get an idea. Okay, I didn't have an idea. I didn't know exactly what I wanted to do. I didn't know exactly what I wanted to be. But at the time I got that idea, that was a turning point for me. And in fact, once I got my idea, I didn't let go until I got there. So today I am a lucky person. I am exactly where I wanted to be. This was my idea of me many years ago. And eventually, a little bit better, maybe less white hairs, but still, I still managed, to kind of get where I wanted to be. A few words about myself so that you know a little bit where I'm coming from, probably some numbers. I'm 41 years old. I know some of you told me that I was 50 something, so no, okay, it's fine. Still getting there. I have two daughters. They were born in Switzerland, so they have double nationality. I'm originally from Italy, probably as you can guess from my name and my accent. I lived in different countries, Germany, United States, Italy, of course, Switzerland. I have one sister and 42 cousins. Why? Because I was born originally in southern Italian family, so it's big stuff, like the big thing. I love history. I think understanding the past is what allows you not to make the same mistake in the future. I love land, so I don't own land. I protect 5 acres of land. That's at least what I think is my mission. And I'm really into, and this is why I'm getting older. But I love production of olive oil. I have olive oil trails. I really like, to get in contact, so to say, with the nature. Something that has always really fascinated me. And this is a little bit more on the personal point of view. On the professional point of view, I've been a scientist for many years. I'm a PhD board certified professional. I have a PhD in vascular medicine, indeed. I published, a number of studies. I had the pleasure and honor to publish into extremely highly recognized impact factor journal when I was very young. But then I switched my career from the basic research to the translational research, and eventually I moved to diagnostics. So I worked in a hospital for many years. I was the head of the molecular pathology division in Basel. I signed a lot of diagnostic cases in my life in the hospital. I also founded a company in the Middle East, in the United Arab Emirates, which I exited. Big mistake. Very big mistake. I exited after 2 years because I was very impatient. And if I would have waited more, probably I wouldn't be here right now. That's okay. So, but anyway, I worked with more than 1,000 colleagues in my career. I was really blessed with the people that I met on my journey. Um, I oversee now a business that it's about $1 billion in size. And as a venture partner, I oversee investment of about $180 million in capital deployment. Um, I'm sharing. I mean, I was asked to make it personal in a way, so I picked some pictures, not to not to share too much about myself, but that was me very young when I started with next generation sequencing. It was definitely something that changed my career. I was lucky. I was in a moment where NGS was moving from being a research tool to a diagnostic tool. So I jumped right into that wave, and I was again one of the first one doing that. So it was a great experience. But I was also like a little bit of a nerdy number person. So I had a lot of these whiteboards with all this crazy stuff written on top, blah blah blah. I still do it right now. I think it's very important to get your flow of idea somewhere. It could either be an electronic board now or whatever, but do it. That's a picture that's very close to my heart. It was my very first plenary lecture many years ago, where I was talking about the value of next generation sequencing in diagnostic, and the room was very full. I didn't expect it that. It was like a huge impact for me. I think I was probably 29 or 30, I don't know exactly. And I was really emotional that day. I couldn't remember anything. And I'm very thankful to the person that did that picture because it's a reminder of a very important moment in my life. In fact, I actually don't know exactly who made that picture. But then starting from there, I started to give talk all around, get interviews. It was like a relatively nice journey of me. So I've been, again, blessed with the opportunity that I got. And most importantly, I started to build a team of people. And one of the things that I learned from my mentor, and I will talk about this one later on, is to try to work with people that know more than you know. In fact, very often I have to tell you, it's not about your own competencies. Probably you don't have that competencies. It's about getting with the right people and together make something that you are not able to do alone, unless you are Elon Musk. I don't know how many Elon Musk are in the room, but it's like so your team, it's one of the most important asset that you will have in your life, whether you're building your own company or working for a corporation. I thought I would also mention this because this is my everyday life, probably hitting the road almost every week. I'm always flying. This is like I think me in China a couple of years ago. Family is very important to me. And so no matter what you're doing in your life, no matter how many flights, investment, whatever you do, company you do not forget during your journey to live as well. So make sure you also have your own life, that it has to be untouched by other stuff. So I was blessed having two daughters. This is my daughter's a few years ago. I always found it a funny picture. And the last thing is I told you I'm passionate about history. So when my father started to lose, to have some sign of dementia a few years ago, before he was completely off, I wanted to write the story of my family. So I started to dig, taking advantage of the team, the 42 cousins, that's why I mentioned it here. We got together and we started to grab picture, history, everything that we could from our parents, our aunt, uncles, whatever. And we actually wrote a book about our family so that this will hopefully stay in the future and, before this memory will disappear somehow. All right. Now, one thing I also want to mention is that you need to have a kind of a plan in a way. And I think the plan also comes from an approach to life and a way to operate things. So at least in my personal experience, I was always told again by my mentor that there's a moment in your life where you have to build. For example, when you are at university or when you're learning, so you have to build your own knowledge, your own competence, knowing that you can't know everything, especially in these days, it's impossible, but we do have tools like AI that can help us. But you need to learn how to learn things. That's very important. So build your personality, build your own profile. That's the first thing. Once you build yourself, you understand what you're good, what you're bad at, start to think about how can you leverage your strengths and try to avoid the places where you have maybe weaknesses. After you think, you can start to act. You can fund your own company, you go work for a certain whatever company, whatever it is. And after you are acting for a certain amount of time, you start to harvest, so to collect the fruits of what you have seeded. And eventually, when you're getting older, like me, you also start to seed something for the next generation. I think it's very important. I have been, again, blessed with people that believed in me. So I think it's also important now for me to seed something for the future. That looks very clean, but that's completely unreal. That doesn't work like that. In fact, in the reality, everything is mixed up. While you're building up yourself, you're also thinking about the fact that I want to build this company. At the same time, you want to rush and do a job that you really like. And at the same time, you want to get the best out of it. Again, my exit in the company was exactly this problem. I wanted to harvest immediately things. And at the same time, you maybe also starting to seed something. So that's fine. It doesn't matter. It doesn't have to be that clean. It doesn't have to be that clear cut. Life is not clear cut. It's in fact, sometimes very complicated and messy. But at least you should have a way of thinking. It doesn't have to be necessarily this one, but sit down, take time, and think about how you're going to get where you want to go. And the first thing probably you should do, and this is what I did myself, they give you a lot of training, as an executive to fundamentally get to know yourself. So for instance, myself, I'm definitely not a genius person, and I met a lot of, Nobel laureate, really person that you can see they are extraordinary, and you realize, okay, I'm an average guy, fine, fair enough. It's okay, but I do have a scientific mind. I did PhD and so on. So I'm kind of structured in this way, and I love to explore things. I get very easily, I get bored when it's like, the usual stuff. So those are some of the things important for me. I'm number driven, but I'm also person driven. I like to talk to people. Probably I talk a little bit too much. This is due to the Italian genes, I guess, but that's all right. I am tenacious, meaning that when I want to have something, I really don't give up. Maybe sometimes I should give up on things, but I don't. I have a relatively good memory, and I like details. I don't get lost into details, but I do like details. I do like to do things, done in a proper way. There will be many other things, but again, this is just a small example of sit down, get to know yourself. You need to know what your strengths are. You need to know what your weaknesses are. And of course, you have to double check this one with the others. So don't just do it, in your own way. Maybe another thing that it's worth mentioning is I heard this so many times, like, follow your passion. You find it in every, career, whatever kind of stuff. Yes, do that, but don't stress out too much about this, So it is important, of course, to follow your passion, especially if you are at the beginning of your journey. I would say it's not exactly the best piece of advice in a way, for a number of reasons. Because to me, it's a little bit naive, saying just follow your passion. Of course, you need to do something that you like, but in fact, at the very beginning, you need to explore things as well. Because if you don't experiment different things, you can never really know what exactly you like. And I think maybe a note for established professionals, as there are in this room, if I may, very respectfully, when you are already an established person, yes, of course, you got to do what you like, because you have to wake up in the morning and really do what you like. I think when you're young, you have the privilege of choosing, but once you are a little bit more into that, I think it's a good idea to do what you like. And in general, I think this is kind of a little bit of an overrated statement. And why is that? Because what is your passion maybe when you are 20, it is not necessarily the same when you are 30, 40, 50, or whatsoever. So we are fundamentally the same, but we actually change every day. So it's important to pick this concept with a little bit of flexibility. Also, I mean, I don't think there's such thing as a perfect job. I love my job, but there are also things that are part of my job that I really do not like. Still, the most important thing is that you like what you do. The reality, probably, I would say, of life is that it is important, once again, to do what you love, but that does not necessarily equal with the fact that you will be always doing things that you like. So that is part of things that you will do that you don't like, but if these are important to make you do what you like, that's still fine. The other thing which I found to be extremely important in my career is networking. Of course, that's also one reason why we're here, and this is a perfect, in fact, event for doing this one. It is one of the top things that you can do to boost your career. I think it's equally important as your competences. Once again, because there will be many people out there that have the same knowledge and competences that you are. So what's the difference? Why is it you and not somebody else? It's also because you probably build a good network. Sometimes it's annoying to hear this one, that you might get into a certain place just because you know the right person. But I want to be very blunt, it's the reality of life. So you better get along with that, if you do wanna go where you wanna go. And one thing is, don't be shy, socialized. So I when I was a little bit younger, I was, oh, should I go and ask this person for this about that, or maybe I should not disturb this person. I actually want to tell you a secret. We are all human. We all wake up in the morning, drink our coffee, do our thing. So even the CEO of my company, just an extraordinary person, I will stop him and ask him for stuff because he's still a human person like we all do. So don't be too much worried about. Don't be shy. Just get along. Go ask. Worst case, you don't get an answer. Worst case, you get a bad answer. Or worst case, they tell you just go somewhere else. But it was a very gentle way of Anyway, the other thing which I think is very important, and you see some picture of me throughout life, is finding a mentor. I was blessed with meeting person. I mean, I had terrible hair, by the way, there many years ago. But I was blessed. I had people that believed in me, that they were in a stage of their life and their career where they were seeding for the future, and I was lucky enough that I was probably some of the seed that they had seeded. And I built very strong relationship throughout the years with these folks. You don't know who they are. You don't need to know who they are. But some of these guys are really, having a huge impact in the world of oncology. I'm not sure they'll be very happy to see this picture in around here, but we're not going to tell them. Now, let's move now to the more thermal stuff. Okay, that was a little bit about the history, and I was telling you before, this that I had this idea and blah blah blah, and that there was this very specific gap that I wanted to fill. What was this gap, and why Thermofisher was for me the place to be to go fill this gap? The gap that I wanted to fill specifically, and that's something that I came up when I was working at the hospital, was the possibility of generating ultra-fast next-generation sequencing testing result. What does it mean? Usually what happens today is that at the beginning of a diagnosis of a suspect neoplasm, or possibly a cancer, most of the time a tissue biopsy is collected. I'm trying to oversimplify this one, just to get you an idea. Now, depending where you are in the world, even here in the UK, actually, let me quote something that's happening here in the UK, it might take over a month before you are getting the result of your diagnosis, including the molecular profiling. If you are lung cancer, if you have an EGFR mutation, BRAF, whatever. And this is really a problem. This is a problem for a number of reasons I'll show you, but mostly it's also a problem for patients and families that have to wait such a long time, with anxiety and what's going on, what's going to be the treatment and so on. And so time in this very specific moment truly matters. But that's not just me saying. I wanted a couple of years ago, and this is published in JCO, which is the official journal of ASCO. We made a study and demonstrate that if you wait too much, if your results are taking too long to be delivered back to the treating physician, this has a negative impact on the survival, so on the clinical outcome of patient. So first of all, we set, by using electronic health record, we set to understand what was the average turnaround time for NGS-based biomarker testing in the United States, because electronic health records are a reality in the United States, not yet in Europe, I would say. And in the United States, it takes about 20 to 23 days to get the result. It's a lot of time, okay? Now, the consequence of this very long waiting is that there's about 30% of patients, so 3 out of 10 patients, will actually start the treatment, most of the time, the classical chemo scheme, without having their molecular profiling result. And the consequence of this is the one that you've seen here. I don't know how much you're familiar with Kaplan-Meier survival curve, But what you see there, the blue line, the group A, those are non-small cell lung cancer patient, which is the most common type of lung cancer, in advanced stage of disease that are treated based on the result of their molecular profiling. And this is their survival, you see that. Now, these other two groups are patients that either started the treatment without having this result, and so they started, for example, a chemo or a chemo immuno-oncology combination, and then once the result were delivered to the physician, they were switched back to what was supposed to be their best treatment approach. As you can see, they have lost quite of an opportunity. They don't manage to get back to the same survival rate as the patient that are treated based on their molecular profiling. And the third group here is the group of patients that will never switch back to a targeted therapy. They will stay on the classical chemo approach. As you can see, unfortunately, look at here, the difference in survival is immense. So that's a problem. There is a gap, okay? So how do we fill this gap? This was for me really the driver. How do we change this one? How do we make sure that molecular diagnostic can be delivered in an ultra-fast way to make sure the most appropriate treatment is chosen for this patient, okay? From an idea to a product. How do we make a product that is filling this gap? And this is how, technologies can really move the needle in a way. So, if that works, that not always technology is working, that's it, but that's fine. Yeah, but this actually was part of the problem. It took some time, okay? The first thing is that we had to lay down, what was the problem and what was the path for resolution. I mean, having an idea per se, it doesn't mean anything. I wanted to have a really detailed, scientifically backed idea. And the first thing I wanted to achieve is, for me, it was important to reduce the laboratory work. A lot of people pipetting stuff, they actually make errors. If you ever worked in a lab, I'm sure you messed up some protocols or whatever. Yeah, that shouldn't work. We are in 2025. We have AI that can tell me in a second how many hairs you have on your head, probably. And how is it possible that we're still pipetting 3 microliter of stuff? No, forget it. We have to change this one. We want to make a system that was fully automatized. There's no man doing anything. And you can do that. And of course, it's also much faster than any other, possible human person. So our goal was to get some kind of, laboratory result within maybe, let's say, 24 hours, okay? The other thing is we wanted to make sure that we would utilize as less material as possible. Because, biopsy, if you've ever seen them, they're pretty tiny things, And I mean, we also wanted to make sure that you could use this kind of technology, for example, for liquid biopsy, which is the other big wave, that it's coming to diagnostic. We wanted to make sure that, the test and applied, with this technology was sufficient to get everything that was clinically meaningful as of, the time where we launched the product. But things, of course, are changing. So we also wanted to make a system that was flexible enough to get updated. You get your iPhone or whatever it is, and you get an update every year or whatever. We wanted to have the same kind of concept because things do not stay the same. We wanted to make sure that this system was working, as much as possible, that the failure rate of this system was very low, and we set certain parameters and thresholds. So you need to understand where you want to go. So the specification, because every time that you fail, there is a patient that is not getting a result, and that's a problem. We also wanted to make sure that, again, the low failure rate would correspond to the reduction of the risk of having to go back to the patient and asking to recollect another biopsy, which is unfortunately what it's happening today. Collect a biopsy, you wait 30 days, you try to generate the result. Oh, it failed because somebody in the lab, I'm looking about you, I don't know if it was you, did you? Failed pipetting something. And then what you have to do, you have to go back to the patient because you used up all the material, and you have to go collect another biopsy, which is a very unpleasant, situation. So no, we don't want to do that. And last but not least, of course, we wanted to speed up the entire consultation problem. You go get your biopsy a day after, you sit down in front of the physician, and you get the treatment, decision. And so in a more simple way, we wanted to go from a very complex, workflow, which is the one that is still out there in many places, to a very simple one. So from here to here, that was the overall idea. And this kind of instrument in my mind, it had to do everything from extraction to library preparation to the sequencing to the data analysis all together in one place. And eventually, after hundreds of millions of dollars of investment and a number of years and hundreds of people working on that, from that idea, hundreds of people, hundreds of R&D scientists all over the world, we ended up having an ultra rapid NGS system. Looks like a fridge. It is, in fact, like a French fridge, but it does everything. You just go there. There's a one pipetting step. You take your sample. So don't mess up the only pipetting step, guys. You can do that. Okay. And after that, you just put it in the system. 5 minutes, click a couple of buttons. It's, well, as we used to say, it's executive proof. So if we can do it, then everyone can do it because it's very simple. You walk away, and 24 hours after, you have also data analysis and full report of your patient. And this actually system, after a few years of testing as a laboratory developed test in many places, and it has been now recently approved just 2 months ago from the FDA. So now it's a full cleared system that can be used for in vitro diagnostic. And again, it can be used not just for oncology, but for a number of things. So, and it was really a long journey to get there. It was years of work from that, original idea on the whiteboard that you have seen to eventually get to this place. By the way, it's called the Ion Torrent Genexus system. And yeah, here you have it. That single pipetting step, it's there. That's all you have to do. And then you go to the system and you put it there. But I think, after this long journey, it's also really important that for me, at least, this is like, okay, now I've achieved something, so to say. I have contributed. Of course, I didn't do this myself with a number of people. It takes a village. I contributed something, but what is now my everyday mission, if you want? And my everyday mission is that what you see in this slide, which I've tried to explain in a minute. How much time do I still have, by the way? 5 minutes? Okay. 10 minutes, great. What you see here in this slide, it's what hopefully will happen everywhere in the world. Now, this is a very special place. This is the Mass Gen Hospital in Boston. It's one most important, of course, institution globally when it comes to oncology. But this is what it's happening now every day at the Mass Gen. So when a patient is showing up at the ER, maybe, with a very compromised clinical situation, maybe this patient is, coughing blood or having problem in respiration and so on. First type of approach, X-ray, MRI, things are done very fast in that case. And eventually you find out, there's a mass in the chest of this patient. You don't know, it could be anything from tuberculosis or to cancer, okay? So what you need to do, you need to go collect a biopsy. In this case, it's fine needle aspirate that you can collect. And now, what they have been able to establish at MassGen and in a number of different places by taking advantage of the technology I just showed you before, is to go from day one and within 24 hours, performing the histological analysis, performing the genotype, in this case, for example, finding out that this patient had an EGFR mutation, which makes it eligible for a tyrosine kinase inhibitor treatment, actually from a company that's based out in here. And then 24 hours after, so day two from the day that the guy is showing up at the hospital, the guy has never even left the hospital. Day two, the treatment is initiated. So the guy goes to sleep the day after he's starting the treatment. Now, the incredible, even though it seems like all very easy, the incredible output of this one is that at 48 days, for example, in this case, you start to see a very clear clinical response for this patient. Now, think about what I told you before. Usually, it takes a month to get the result, but then you have to start the treatment. So usually, other patients, they start treatment at day 40 something. This guy is responding already to the treatment at day 40. It makes a huge difference. this is going to change the clinical outcome of this patient, right? Of course, this is just one example. I mean, the citation was a big deal, published as well in JCO. But the point is that this is now up to the people working in the institution to establish a strong workflow that makes this happen if that works. But what is my job now? Probably I should retire. I did this. I did this thing, so everything is done. Whatever. But what is next for me? So what do I want to achieve? What is my kind of next goal, if that ever works? To get to my next probably goal, it does take some time. Fantastic. That's what I wanted to achieve. And this is one of the things that you do when you work and the privilege that you have when you work for a large company like Thermofisher. You can scale. So what we have done in the last few years after we eventually released this new technology, we scale it up. And now you have over 1,250 clinical laboratories all over the world that are using this technology. It's no longer just at the MassGen. It's pretty much in everywhere in the world. Of course, the adoption is different in certain places compared to the others. There's a lot of things that we still have to do. But to give you a number, we estimated that in 2024, so last year, 850,000 cancer samples were tested using the ion torrent technology, which to me, it's an incredible number. You remember I told you that when I was the head of the molecular pathology division in Basel, I was signing off about 4,500 cases a year. Now the impact that I have, it's no longer on 4,000 patients, it's on 850,000 patients every year. And this is a number that, of course, will continue to grow. That's the reason why I wake up in the morning, to make sure that everyone has access to this kind of technology. That's the kind of impact that is driving me every day. Sorry. Oh, thank you very much. Thank you, Jens. And from the moment that again we launched this technology, another of the things that I do is partnership with pharma. This is part of my team and other teams within my division that we're doing. Of course, to make sure that we connect the right patient with the right therapy at the right time, you need to get along also with the drug producers. And here you see some of the partnership that we have established in the last few years. But this is a ever-growing list of new drugs that now, thanks to this technology, can be linked to the right patient. One thing that I do want to mention, and this is another of the things that my team is taking care of, it's demonstrating additional application and also expanding the reach. We started with solid tumor, and now we also wanted to go into the hematology field. And our technology has been recently chosen as the technology for the NCI MyeloMatch clinical trial. Sorry. Oh, after 30 minutes, I start to slow down. But it's this is a unique clinical trials. In case you don't know about this one, is the largest ever founded trial by the NCI in the context of hematology, which unfortunately is a little bit in disgrace now because, as you know, NCI and NIH will face a huge budget cut next year, but still the project managed to kick. And just to get you an idea of what this is, and again, what is my team doing, this is a clinical trials that span entire North America. There are 2,200 different hospitals and institutions that are participating into this trial. The whole concept into this trial, which has 27 different arms, which is amazing. It's incredible. It's to match the right patient again with the right therapy. But in AML, which is the disease that we are trying to tackle here, time is everything. And so no matter where you are from any of these dots in here, within 72 hours, you need to link the patient to the right therapy. The only way to make such kind of trial a reality is to have a technology that it's super ultra fast to do the molecular profiling. And that's why our technology was chosen for this one. So with this, I want to get to the conclusion. And the thing is that, again, really, what I told you in the very beginning, do make sure that you align with the mission of the company that you will either build yourself or that you will want to work with. But also, I do want to mention that in case you are interested, of course, in Thermofisher, there's a number of things that we do. We span all the way from supporting healthcare with clinical diagnostic and many other different fields, to the life science, to the applied market, to the pharma and the biotech. So again, we do all the way from laboratory equipment to chemicals and to digital solution, if you want. And in case you are interested in R&D, in case you are a scientist, as I told you, we have over 7,000 scientists working with us all over the globe. And we do have many different sites, a little bit, all over the places where we have center of excellence from everything from mass spectrometry to sequencing to atomic imaging and clinical trials and anything else you can probably imagine in the life science business. So reach out. If this is one of the things that you want to do, have a look at that. We're always, of course, looking for talents. And with this, I actually added one slide yesterday evening, which I was very puzzled, and it's going to be my last slide. I wasn't sure whether I had to add it or not. This is a slide which will make half of the people in this room very annoyed, and the other half of the people very disappointed. Okay? You ready for that? All right. It's not this one. Sorry, this is again the Thermofisher. I apologize. It was not a marketing trick. No, just again, I think it's important you understand that we do span from life sciences solution to specialty diagnostic, analytical instrument, laboratory product, and biopharma services. Is what I told you before, but in a more structured way. Okay, now you can get disappointed. There you go. Not this one. Again. This is once again to remind everybody our mission, which is to make the world healthier, cleaner, and safer. And okay, the corporate one is ticked now. I can finally disappoint you. That's the slide that is going to disappoint you. I leave it there for a couple of seconds to digest it. So what do you see? So what I see is myself 20 years ago, there. And this is me a couple months ago. That was my very first car, the first car I bought with my own money when I moved to Germany, 150 euro. Lancia Y, 1992. It didn't have a mirror. It was a car used for delivery and other stuff. It was a fantastic car. But that's where I started, in a way. Now, why am I telling you that I'm going to disappoint you? Because half of the people in the room are thinking that 20 years ago, my goal was to go from there to this one here. And that's not true. I never had in my mind that 20 years ago, after, I wanted to be seated in a Maserati GT, in case we can talk about it. I'm very much into cars, as you probably realize. But anyway, no, that was never my goal. And if your goal is to go, it doesn't have to necessarily be that scrappy car in the beginning, but if your goal is to go from there to there, probably you are on the wrong path. My goal 20 years ago was to fill a gap. This is nothing but the consequence, the benefit of me filling that gap. I never pictured myself sitting in a Maserati GT, which is a very comfortable and beautiful car. But let's not talk about it. It was never about this. It was always about the mission. Me waking up in the morning and doing something that I really believed could make a difference. That was always, always, always about there. Once again, this is nothing but just an additional benefit. It's very nice to have, but it was never my goal. And again, if you are just focused about this one, you're gonna get lost through the journey. If you're not focusing about this, but you are focusing about your mission, you have much better chances to sit it in that car. Thank you very much.
We're going to start the Q&A now.
I have what? The Q&A. The Q&A, please.
Hi, thank you for your talk. I'm Dennis from Norway. We actually have a site, Thermofisher site, doing Dynabits and T cells therapy now in Norway, Oslo. I was going to ask you about in the beginning when you started about mentoring. I really love that you bring that up, and I think it's very important. And I do some mentoring myself, and looking for mentors as well. Can you double click on the importance of the mentoring in your journey and how you approach it? You know, like know-hows.
Yeah, absolutely. I'll try to be brief, otherwise this is another talk. But one of the first, one of my first mentor, that guy with incredible fancy hair, that's why I also had that kind of 80s style hair. He was a guy, he was a very famous professor of biology at the university where I studied originally in Italy. He was a guy that was NIH, very, very important guy. And I remember, showing up at his lab and say, I want to do my bachelor thesis with you. And the guy is looking at me and said, I'm not interested. Okay, just, okay, that's a problem. So then, I went out and then I realized, no, but I really want to do a bachelor thesis with this guy. So I went again and the guy very politely, said, I know I don't really take any other students, I'm not really interested in this. So basically it took me a month showing up at his office in the morning, and it's a true story, every single day, not always at the same time, because in Italy times is a floating concept. But anyway, every day in the morning when he had a beautiful motorbike at that time, he was coming with his motorbike, and the first thing you would see in front of his door, it would be me standing there, always asking for the same thing. Eventually, I got to his lab and I did a bachelor thesis, which was the beginning of my career. So don't give up. And again, do talk to the people. I was lucky. I probably was very annoyed by me. So in the end, they said, okay, just do it. And, whatever. But again, I got what I wanted, and it was an important moment for me. Now, if I translate this to myself, I always also think that, maybe, should I take just another, training person? It's extra work. And if you have to do it, you have to do it right. I have to invest time in people. But then again, I always remember what happened to me and I say, yes, I have to do it because that's exactly what happened to me. Somebody that couldn't care less about me believed in me no matter what, and, started my career. So that's the kind of same approach that I have. 15 minutes. Thank you. And thank you for your question.
Hey, Luca, my name is Sergi. Thank you so much for the talk. Super engaging. You definitely tick all Italian cliches.
Including the tie, probably.
And so maybe I missed it in the presentation, but my question is, when you identified this gap, you were passionate about this problem. Um, were you already, you were not yet in Thermofisher, right? So when do you approach Thermofisher? Or was it already that you were working with them afterwards and you remembered about this gap and then kind of have a power position where you could work on this solution? So how was that?
It's an absolutely great question. I apologize if I wasn't clear. No, I was not working at Thermofisher at that time. I was still in Basel, the head of the molecular pathology division. But I was a key opinion leader, what they say. So, I was very often going to conferences, presenting results. So I was a relatively well-known person in the field of molecular pathology at that time. And I worked with all the big companies, as an advisor. And then eventually I decided to specifically go to Thermofisher because of the alignment between my, let's call it values in this case, and mission, and the fact that I found there a fertile soil to seed this idea. So I went there and said, look, guys, I have a problem. It takes me a month to generate this result. And it's just unacceptable because when I show up at the tumor board to talk about this result, sometimes the physician, the treating oncologist say, actually the patient is gone, which is terrible. Then your day is completely gone. You're devastated. You say, I didn't even manage to generate the result for this guy. So in fact, I went to Thermofisher executive and started the conversation at that time. Of course, there was some awareness about the need of generating something like this one. But in the very beginning, I think I was important to lay down that slides. What exactly do we need to address? That's really something like, I think the slide that I presented to Thermo leadership at that time. That's what needs to be done. These are all the boxes that this technology needs to tick. And again, I was lucky because on the other side, there were people with the right mindset to hear what I was saying.
Hi, Luca. Philip from MGI. So I guess a question everyone wants to know about. So why Maserati, right?
When?
Why Maserati? Why not others? But no, actually, the serious question is, so you breached this gap that existed, and what's our next gap? What should we fix in this field?
Excellent question. Look, what it's happening today is that, again, these and now there are other technologies that are very fast in sequencing. There's a lot of opportunities out there. The field has really moved forward, made almost a leap in the last 10 years. But one of the big issues that we still have today is that it's not always possible, for example, to do a diagnosis based on tissues, because sometimes the anatomical place where you have to go and collect the tissue is unfeasible, maybe because this patient is already metastatic side, and where would you go if you have 10 different metastases? It doesn't make any sense. So one of the gaps that needs to be filled is possibility of using other kind of starting material, including liquid biopsy, CSS fluid. So taking advantage of other routes, so to say, to get to the same result. It is happening. I was a big proponent of liquid biopsy. I had the honor to sign off the very first liquid biopsy case diagnosis in Switzerland 10 years ago, I think now. And I was going around the world and say, liquid biopsy is going to take off. It's going to real. It didn't happen yet after 10 years. So we are not yet there. It's a big gap. It still needs to happen.
Thank you for a great talk. My question is kind of similar to a previous one, but I was just wondering, when you had this idea, what made you want to go to Thermo, which was more established, rather than taking the idea yourself and like taking a more entrepreneurial approach?
Great question again. And I mean, of course, I had kind of the idea, I'm going to do this myself. And but I mean, now I am in a venture capital, also as a venture partner. At that time, I knew nothing. I was in academia, and there weren't many of these, events like this one now, where I think you can get a lot of knowledge how to get the connection. It was many years ago, 10 minutes. It was many years ago, and I didn't know what to do, to be honest. So I was scrambling around, and I realized that such a project would take, millions and millions of dollars to make it happen. And I probably at that time established that even doing a startup would have been kind of possible, but would have taken a lot of time. Now, one thing, maybe please one moment to reflection is this one. If you want to do something, of course, then go make a startup, but it's going to take you a certain amount of time that you just have to invest to collect capitals, explain the idea, blah, blah, blah. When you go to a large company like this one, the money is already there. Capital can be deployed much easier. And so there was much less time, not lost. It was lost maybe for me. If I would have founded a company doing this one, I would have been probably billionaire by now. But I wanted to have the fastest path to make that idea something real. And so I went to somebody like Thermo that already had the capital to be deployed. Thermo didn't have to ask anybody. They had to just make a decision themselves. Executives sit it down and say, okay, how much money do we need? Blah, blah, blah. What's the business case? Blah, blah, blah. We do it. Okay, we do it. That was the major difference. But again, it's probably is not the only way. I could have gone down the way of startups and this kind of stuff, but I just wanted to get to the point, and that's why I went to a large corporation like Thermo.
Thanks, Lucas, for such a fun and inspiring talk. I really like it. So I'm Sensen from Mauritius. So I was wondering if you had a time, at the lowest point in your career where you faced a challenge, but it didn't go as well as it should be. Like, what did you do and how did you go about with it?
Thank you. It's a great question. I mean, I failed as many times as you can possibly imagine. Many times. One thing that just definitely It was during my PhD. I think it was about a year after my PhD. My PhD supervisor, which eventually turned out a big friend of mine, you know, is calling me into this office after 1 year and says, Luca, look, I don't think we're going in the right direction. I mean, the experiments are not really working. I think you should put more effort into this. And yeah, guys, I was really working a lot at that time. I get that. I was really working a lot. He said, this was Heidelberg. It's like Cambridge. It was one of the, they, of course, rightfully, but this is like the top-notch place. So it was expected of me, to do that. But still, the experiment were not going in the right direction. And so I go out from that office. I almost wanted to say, I'm going to give up. Maybe PhD is not for me. I don't have the strengths. I'm not good enough. I don't manage to do this. And what has really changed for me was my family. So at that time, I was with this fantastic girl that eventually turned out to be my wife. And I think also talking to my family was a major difference. So I think they gave me the strength that I didn't have. And they said, no, you're going to make it. You can't give up. You invested too much in this one. So I think they gave me that kind of push. So the answer would be, sometimes you don't have it inside yourself. You need to find help from the people that are around you. That's why, family has been always very important to me.
Thank you so much, Luca. I'm Eleni from Greece. I was wondering if you could share one of the best pieces of advice you have received.
Best piece of advice. I received many of them, but probably one. I mean, and from multiple of my mentors, the same thing I heard. It doesn't matter if you're gonna achieve something, whatever you're gonna achieve in your life, whatever. Just always be very humble. And it's not always the case, I can tell you. Sometimes we were discussing about this yesterday somehow. Especially when it's called the middle of the sandwich. So like, in not the starting position, not the executive, like in the middle of the sandwich, you do have a lot of people that are quite arrogant. This is the main reason why they're not gonna go one level up into the upper part of the sandwich, if you want, because they think a lot about themselves. And before, your question, I hope you appreciate that. I say, I'm just another dude. Never forget about this one. There are many other people, out there that have done probably the same thing and much better than what I did. So I think always stay humble. It's gonna pay off.
Thank you, Luke. My name is Akim. I'm from Uganda. I run a startup and we developed a diagnostic device for malaria. One of the biggest challenge I see from the global south is access to the patients. And for my own startup, when we developed our first prototype, we had cracked the tag. The device was able to detect the disease, but then accessing the people, because you have to deal with infrastructure, human resource, and stuff like that. So my question is, how do we bridge that gap to reach to the patients in the remote communities? Because I feel like that's where in my own perspective, from my background coming from the continent, that's where the biggest gap is at.
Yeah, thank you very much. It's a great question, and I think if I have to say, it's one of the things I'm not satisfied yet. It's access. I made you the example of the mass gen, but let's be honest, at mass gen, it's easy, right? I now live in Switzerland, blah blah blah. It's easy, they can go get this device tomorrow. But that's not the point. The point is to making sure that no matter where you are in the world, and that's why I said there's still a lot of work that needs to be done, this kind of technology needs to be accessible, especially in complex geographies. Like we do now have a project in Australia, for example, where sometimes some communities are extremely remote, and where we're going to leverage liquid biopsy, because again, you can go to the patient and get a biopsy, in the middle of nowhere, in Australia, but you can collect blood pretty much everywhere. So that's one of the projects. So to be fairly honest with you, sometimes, let's put it in this way, the business case for such things do not exist. They do not hold. And so not every company is interested in investing in that thing, because if you look purely and only at the numbers, the answer is no. I'm not going to do it because there's no really the right return on investment. But I think the step you have to do is to let them understand, and there are companies out there, and Thermofisher is one of these, to be honest. Sometimes the ROI is not necessarily in dollars. There's other kind of ROIs, which is social impact, and it is valuable and it is valued. So I think in your specific concepts, maybe you should go try to get resources and talk to people that do not necessarily, even angel investors and some of the VC are backed up by people that don't necessarily want to make money as a return, but they do want to have a social impact as a return. So you go need to talk to these people probably in this case. Two minutes.
I'll keep it short. Thank you for the great presentation.
Okay.
A very quick question. We talk a lot about grit and how important it's to be resilient, so perseverance, and basically not take no for an answer as you did. But what is the fine balance, and how did you find it in actually realizing that maybe sometimes we need to divert and do something else and accept failure? Like, what is the fine balance, and how did you find it?
It's an excellent questions, and I believe again has been one of my weaknesses, because sometimes I'm really I want to get something and I do get a no, and I do get a no, and I do get a no. I think when you start to bleed, you probably have to realize that, okay, that was really a no, and I have to go try to do something else. Now, I have not been very good in finding the balance, to be fairly honest with you. So I'm not in the position of giving you. So I took a lot of slap in the face of no with some of the ideas, and the answer is that I don't have an answer. I'm sorry. It's going to be my last answer for today. But I don't know. It's all very personal, I think. It's and then there is no perfect formula to say when you have to stop. Sometimes you will realize on time. Sometimes you're just going to crash in the wall, and that's going to be painful. That's where you realize you have to stop.
Thank you very much.
I just want again, sorry for 1 10 seconds. I want to thank again the organizer. Thank you. Thank you, Ioannis. Thank you, everyone.